Well, this is the perspective. So I think it's about time that we started talking about PRRS again. Any swine veterinarian will know that you can't go more than maybe 30 minutes in any conversation before talking about PRRS. And so we're going to go back to our old friend, and I'm not referring to you, Enric, but our old friend PRRS and our old friend Enric Mateu. Professor Mateu is in the Department of Animal Health and Anatomy in the Universidad Autónoma de Barcelona, and he also works in CReSA as a researcher, and is, a real world authority in PRRS immunology, and in my mind combines both a strong academic background with a really good practical understanding of PRRS. So I'm really looking forward to this very important topic, which is recombination in PRRS, how it happens, and how we can cope with it. And Enric, on this really, I think it's a challenging topic. It'd be great to hear your presentation. Welcome to the perspective.
Thank you very much. And first of all, I would like to say that I expect to be more friendly than the virus. so under this point of view, I expect the talk to be very friendly. Certainly, this is one of the hot topics in PRRS nowadays, and this is why, and it has to be mentioned that because of some events of recombination that happened these last days. First of all, a brief outline of the talk. I will talk a little bit about the concept of recombination, what is recombination, how frequent is recombination in animal viruses, and how this recombination happens, particularly in RNA viruses that are the most common agents of recombination. Point number 4 of my talk will be why recombination happens, and this is a little tricky question because indeed recombination in nature is not clear why it happens, what is the meaning of this recombination. What we will know in this talk after that is the main factors and constraints driving the occurrence of recombination. We can recognize several elements that are driving recombination and that we can use to stop it. Some practical facts about recombination in PRRS, things that are true and things that are not true, and a very important point, some good practices to limit the occurrence of recombination in the field. And finally, the need for surveillance. How can it be done? What is the importance of doing this surveillance of PRRS in the field? After this part of the talk, I will have a very short slide of take-home messages. So first thing first, and the first point here is about what is recombination. If we take a very, let's say, academic definition of recombination, we can say that recombination is the exchange of genetic material between different organisms. And this exchange of genetic material results in a progeny that harbors part of the genome from different individuals. So that's a very simple idea. When we talk about recombination, we talk about exchanging of genetic material. However, when we talk in biology of recombination, indeed we are talking of two very different things. One is recombination, stricto sensu, that is the exchange of genetic material that happens within a continuous genome. And the other is reassortment. And reassortment is the phenomenon that happens with viruses that have segmented genomes. This would be the case of influenza or rotaviruses. In those cases, one or more segments of the genome are exchanged, and this is the way in which new pandemic influenza strains are usually generated. In the case of PRRS, since we have a linear genome, what we are talking about is about recombination, stricto sensu, in strict sense. How frequent is recombination in animal viruses? As we saw in a previous talk, recombination happens and happens very often, but it doesn't happen with the same frequency in all viruses. If we look to retroviruses have the highest recombination frequency that we know for all viruses. the recombination frequency in retroviruses is 10 to the minus 4 to the 10 to the minus 5 per site per generation. What that means? That means that in practical terms, virtually recombination happens in every viral generation in the body. If we think that retroviruses, for example, AIDS virus or feline immunodeficiency virus, for example, they are replicating to the levels of 10 to the 8, 10 to the 9. They have very long periods of replication. In practical terms, that means that every progeny contains recombinants between the different viruses, the different variants that are circulating in the animal. With a slightly lower frequency, 10 to the minus 5 to the 10 to the minus 8, we have most of positive sense RNA viruses. And that means that recombination happens frequently. And this frequently means that we can find often recombinant strains. This happens mostly with coronaviruses, with picornaviruses, with arteriviruses, but we can also find recombination in other type of RNA viruses. At a lower frequency, we have recombination in double-stranded DNA viruses. It's quite common between the double-stranded DNA within the DNA viruses. It's much less common in single-stranded RNA viruses. Recombination through reassortment can be common, but recombination itself is a very rare, very infrequent event in negative sense RNA viruses. And after all these other groups, the single-stranded DNA viruses are the ones that are known to recombine the less. So in other words, when we talk about PRRS, we are on the top. We are within the range of very frequent recombination events. How it happens, this recombination in RNA viruses? As a matter of fact, we know at least 3 different methods of recombination in RNA viruses. But in practical terms, the most common mechanism by which this recombination happens is what we call template switching. What is this template switching? These RNA viruses are being replicated by the intervention of an RNA polymerase. This RNA polymerase uses the original RNA genome of the virus to produce copies of it. Normally, and this is what we have in the slide, in the upper part of the slide, we have a nascent strand that is being copied from the original one. But sometimes this polymerase disassociates from the template. And soon afterwards, the polymerase associates to another template. If the template is the same, that is, we have a homologous template, recombination may be produced something that is viable. If the template to which the polymerase associates again is not the same, we will have a non-viable product. Let's see that. First option, the polymerase associates, for example, to cellular RNA, messenger RNA. The resulting product, that is half genome of the virus, half a segment of a cellular RNA, normally is non-viable, and this will be degraded afterwards. This is very common. In a second case, what can happen is that the polymerase associates again with the viral genome, but not in the same position, a little bit forward, a little bit backward from the original point. This will create an insertion or a deletion, and depending on the place in which this insertion or deletion takes place, the resulting virus can be viable or not. Or maybe, after disassociating from the genome, can go back to the same place, and this will produce a viable recombinant. It contains pieces of 2 different genomes, but they were continuously replicated, so this genome is fully viable. Or maybe there is a different position in the genome where the new replication starts, and again we have an insertion or a deletion. So as far as we can see, of all the potential recombination events, most of them will produce non-viable copies of the genome. So to understand well that, and this is very important, this type of recombination is what we call a replicative recombination. In other words, it only happens during the replication of the viral genome. So, first very important point, we need replication to have recombination. And the more the replication, the higher the levels of the replication, the more easily this recombination will happen. And second point, we saw it that since this template switching is not governed by nothing else than a random event, this recombination can produce non-viable viruses. That means that probably we have hotspots in the genome, that is, places where recombination may happen just because the progeny resulting from recombination at that spot is still viable. Why recombination happens in positive strand RNA viruses? This is a very tricky question because the whys sometimes in evolution are not very meaningful. They happen just because it's one of the ways in which the evolution finds a path. But we can explain some things about RNA viruses. The first one is that positive-stranded RNA viruses have two common characteristics. They exist in very large populations and they have a very high genetic diversity. And the main reason for that is thought to be the effect that the viral RNA-dependent RNA polymerase has relatively low fidelity. In other words, about every 10,000 nucleotides that are incorporated in a copy of the genome, the polymerase makes a mistake. So that means that the accumulation of mutants within an individual, within the population of viruses, is very high. We can see it this way. We have one viral particle, the black dot at the left, extreme left part of the picture, and in the next generation, when this virus replicates, the progeny of this virus will contain some mutations, just because this random effect, because of this random error of the polymerase. These two copies of the original virus will replicate again, and the progeny will contain as well some mutants. So in other words, after a number of cycles, the whole population will be variants, will be mutants compared to the original virus. In the infected individual, so the virus exists as a quasi-species. The quasi-species is this cloud of closely related variants of a viral genome, what we call the mutant spectrum, that compete and interact. Each mutant may have a different fitness for replication in the individual. And finally, when we reach the equilibrium, what we have is the best combination, the fittest combination of mutants in this cloud. If we understand that from an evolutionary point of view, there are several reasons that may explain why recombination is so frequent in these viruses. The first one is because this recombination may introduce advantageous gene combinations. In other words, by recombining, we can have variants of increased fitness, variants that escape the immune system, or variants that can produce a species jump. The second reason may be because recombination may help to reduce the accumulation of deleterious mutations. It's easy to understand that if the virus is accumulating mutation after mutation, some mutations will be neutral, but some other mutations can be deleterious. If there were no repair system for that, finally the whole population could accumulate a lot of negative or deleterious mutations. By recombining with fittest or with the fittest variants in the animal, we can produce new variants that are fittest as well. The third reason that we can have for that is just because recombination can be understood as a general mechanism of repair, not just to repair the deleterious mutations, but the general repair of the genome. Or a very simple explanation, maybe, and just maybe, recombination can be a byproduct of the replicative strategy of these viruses. In other words, no particular purpose of recombination, just a thing that happens because of the methods that use the virus for replication. In any case, it doesn't matter very much at this point what is the final purpose in evolutive terms of recombination, but we know very well what are the main factors and constraints driving the occurrence of recombination. First point, and this is the key one, recombination requires a co-infection of the target cell. We cannot have recombination if we don't have two different variants of the virus within the same cell. And this is very important in PRRS because when we talk about recombination, we are saying that the animal is infected by two different variants at the same time. However, this is not the teaching, because we know that for some viruses exists a phenomenon that we call viral interference that could block co-infection. What this viral interference is, we talk about viral interference when the infection with one variant avoids the infection by a second one. This is known, for example, for some human viruses. The second point that influences the chance of recombination is whether both parental genomes are equally or differently frequent within itself. And this is very easy to understand from an intuitive point of view. Let's imagine that we have one cell that is infected by two viruses. One of the viruses is replicating very efficiently to a very high level, and the other one is replicating less. The chances for the most common to recombine with itself are higher than the chances of this highly efficient to recombine with the other. Or even the chances for the most efficient in terms of replication being the measured parental are higher than the chances for the other that is replicating less. So the levels of replication matter. It's not the same if the virus is replicating to a very high level or not. We saw it before. When we have recombination, the products can be viable or not. So the closer the two variants that are replicating, the more likely the recombination will happen. In the case of PRRS, we know very well that type 1 PRRS may recombine with type 1 PRRS, and the type 2 or species 2 of PRRS may recombine with the species 2. But we don't have any evidence of type 1 and type 2 recombining between them. And the reason is in this slide. The farther one to the other, the more likely the products will not be viable or will contain insertions or deletions that will produce non-viable or poor-fitted variants. These variants will be removed. This is also a key point. The progeny of this recombination has to be viable. And when we think of the PRRS genome, this is not the teasing. This is the PRRS genome. The PRRS genome is made mostly 3/4 of the genome of the non-structural proteins, of the segments encoding for the non-structural proteins. And then we have 1/4 of the genome that encodes the structural proteins. As you can see in the slide, for the structural proteins, most of the ORs of the open reading frames, the segments that are encoding the protein, are lapping. So that means, for example, that if you modify ORF2, probably you're affecting 2A and 2B. So a recombination may have an impact into different proteins. This is also, again, very unlikely to produce a viable progeny because when you have such a small genome, are very little the parts of that genome that can be removed or modified. Again, this suggests that there should be some hotspots for recombination. In other words, some parts where the changes produced by recombination are not affecting the viability of the virus. A fourth element for understanding recombination is the duration of the infection. And as a matter of fact, the longer the infection period, the higher the chances for having a recombination. If we look at the different models of infection, of viral infection, the most common one is what we call in the upper part of the graph, the acute self-limited infection. The animal is infected, suffer the infection, the virus replicates in the body for, let's say, 1 week, 10 days, and then the immune system eliminates the infection. The chances for recombination are very small because this animal needs to be co-infected at the same time by another variant. If the period of infection is very short, the likelihood of a recombination happening is very small. But in the case of PRRS, we have a completely different pattern. In PRRS, we have a viremia that may last, let's say, 21, 28, 35 days, and after the cease of the viremia, we still have infection in the lymphoid tissue. And this infection in the lymphoid tissue may persist for weeks or even months. So here we have a good model for recombination if co-infection occurs, particularly during the viremia. So summarizing, for recombination taking place, and obviously for the progeny spreading, it must happen that, first, a co-infection by two strains or variants happens in the same cell, that means in the same animal. B, replication of both variants happens at significant levels. Both variants are replicating enough to having a chance for recombining. C, the progeny of such a recombination must be viable. And D, this progeny must be fit to be transmitted and replicated. If this progeny cannot be transmitted, the recombinant will exist but will fade out from the farm. And finally, this new variant created by recombination must not be outcompeted by other variants. In other words, maybe you can exist as a recombinant, but if there are other variants that are fitter than you will disappear from the population. So, up to this point, two very basic ideas. Recombination is a natural phenomenon, and this is intrinsic to the evolution of the positive sense RNA viruses. And two, recombination is a matter of probability. And that means that our actions may help to increase or to decrease such probability. First question for now. Recombination will probably happen more frequently, A, between vaccine strains, B, between wild-type viruses, or C, between wild-type and vaccine strains. Let's think a little bit about that and give your answer, please. Remember the elements that are driving or that are constraints of these recombination events. Let's see, 100% say between wild-type viruses, and the answer is between wild-type viruses. And the answer is easy. Since wild-type viruses are replicating to higher levels, the chances of producing recombinant viruses between this type of viruses is higher than between vaccine virus and wild-type virus or between vaccine strains. Let's move a little bit on. And let's see some facts about recombination first. First idea, recombination is probably very common between wild-type PRRS. In this slide, you see a picture depicting the genome of several strains of PRRS1. And the basic idea is very simple. If you see one strain that is in the same color for the whole genome, from 1 to 11, like, for example, the 3 red ones that you have at the bottom of the picture, that means that all genome segments can be identified as being originated in a single variant. When you have a genome with 2 or more colors, that means that that genome is made of several pieces of different parentals. This is what we call a mosaic, a genome that is made of different pieces. Let's look, for example, at the number 4 starting from the bottom. Okay? You see we have a piece that is black, then one that is white, another one that is black, one that is red, 2 that are white, one that is blue, another that is white, orange, gray, and white. So at least we have one parental that was of the black type, another parental that was of the white type, another parental of the red type, another parental of blue type, one orange, and one gray. We can identify at least 6 events of recombination for that strain. Very simple idea. Recombination happens just because animals are being infected continuously in our farms. If we take a comparison of what happens with PRRS, the recombination rates for PRRS are similar to that of coronaviruses. Some other facts. Recombination probably happens between closely related isolates, but also between more distant ones. In this picture, you can see 3 trees. Tree in A shows the phylogenetic grouping of several isolates in Europe. You can identify subtype 1, what we call Western Europe PRRS, subtype 2, subtype 3, and subtype 4. And these 2, 3, and 4 come from Eastern Europe. If you look at B, for example, when we analyze deeper this genome, we can see that the strains belonging to the 4, the 2, and the 3 subtype can be in the same group. This is a clear indication of recombination between different subtypes. So, up to now, we can identify recombination only between 1 or between 2, but yes, we can see recombination between distant isolates within 1 or within 2. Recombination may happen also between field and attenuated vaccine strains, and this is a very proof of that. This happened in China some years ago, and they identify one strain that they call EM2007. This strain EM2007 was made, you can see, for example, in the first tree, upper tree at the left, was made from a big part of the genome of the strain CEH1R. This was a live vaccine used in China, but also contained parts of the genome of the strain Wuhan number 1. This strain Wuhan number 1 was a highly pathogenic one. In other words, one live vaccine that was in use in China, not in Europe or in any other part of the world, recombined with one field strain of high pathogenicity. And this tells us that this recombination may happen. The third point about recombination in PRRS is the possibility of recombination between attenuated strains as well. And we have several studies showing that recombination is also possible between MOBs of the same species, between PRRS1 or PRRS2. We have 2 recent publications showing that in Europe. In all cases, this recombination happened because both vaccines were used at the same time or one immediately after the other in the farm. The impact of such recombinants between vaccine strains or vaccine and wild-type viruses is variable, from very little compared to the previous situation to high when recombinants are introduced in native animals. At present, we cannot forecast these. So a recombinant may fade out from the farm because of poor fitness or may predominate in the farm. A recombinant may be more attenuated or more violent. And at present, we cannot forecast the characteristics of a recombinant based solely on the sequence. So the very important thing is how to control the generation of these recombinants. And a very simple reminding, for recombination taking place and for this progeny spreading, it must happen. Co-infection, replication of both variants, recombination must produce a viable progeny. The progeny must transmit and replicate, and this progeny must not to be outcompeted. We can control very well co-infections and replication, and the other 3 points are out of our control. A new question: to limit recombination of wild-type virus with vaccine strains, your best approach would be A, to avoid blanket vaccination of sows, B, to ensure that winners are not viremic before vaccinating, or C, to vaccinate piglets as early in life as possible, 1 or 2 weeks of age. Think a little bit about that and put your vote. Blanket vaccination, that means vaccinating all the cells at the same time, avoiding vaccination of viremic piglets or vaccinating piglets as early as possible in life? What's your opinion? 100% to ensure that guineas are not viremic before vaccinating them, and the right answer is to avoid vaccination of viremic piglets. So you are very good at that, 100% success. So what we can do? The first thing that we have to realize is that controlling PRRS without vaccines is extremely difficult. So we have to take full advantage of vaccination without creating the problem of recombination with vaccines. The first goal we have to keep in mind is to reduce the chances for co-infection. 4 very basic points. Do not vaccinate viremic animals. That means check regularly when pigs and goats. Do not use 2 different vaccines, live vaccines at the same time. It's very simple. Implement a 1 MLB at a time policy. Do not use 2 different MLBs, one after the other, immediately. That is, establish a washout period. And increase biosecurity for reducing lateral introductions. And one important point here is how long it takes for this washout period. This work was done, this picture that I'm showing you, corresponds to a work done by Javier Martinez Lobo in Madrid, and they examined the duration of viremia in very young piglets, naive piglets, after vaccination with different vaccines. And this shows you that in this model, and I have to say that this is the most susceptible model that we can use, naive very young piglets, viremia is about 21 or more days. So a reasonable washout period between 2 different vaccinations would be at least beyond these 21 days, I would say more than a month. We can reduce viral replication. And how can we reduce viral replication? First point, do not use vaccination in naive herds. If your sows are naive, do not use a live vaccine on them. If the farm is endemic, keep strictly the vaccination program of sows. Having immune sows will reduce viremia if they are infected and will increase the transfer of maternally derived antibodies, limiting thus the transmission among piglets. If piglets are vaccinated, check for blocking because of maternally derived antibodies and check efficacy of your vaccination. And check regularly the viral circulation dynamics, so have profiles in your farm. And going for the last point, the need for surveillance, how can it be done? We need to know how frequent is this recombination in PRRS, and we really don't know. So one thing that we have to do is regular sequencing, but not just OR5 genome. We don't want to know at this point the epidemiological trace. We want to know if this recombination happens, so we need full genome sequencing. We need to screen the virus when incidence is changing the pattern in vaccinated farms. When something happens in the nurseries or in the maternities, check it. We have to screen the farms to confirm washout when we are changing MLB vaccines. And again, we have to sequence whole genome if outbreaks are seen in vaccinated farms. What is happening in these farms? To that, we can add the random sequencing of field isolates to understand how frequent is this recombination. Ending, some take-home messages. Number 1, recombination is a natural phenomenon that participates in the evolution of single-stranded RNA viruses. Recombination requires co-infection, and this may involve 2 wild-type strains, 2 vaccines, or 1 of each type. The result of recombination can be not forecasted, but the possibility of generating a strain with high fitness of transmission exists, so we have to control it. We have to use vaccines judiciously. We have to define good practices to reduce the frequency of recombination. A very simple one, do not use 2 different MLBs at the same time, or one immediately after the other without respecting washing times. And in vaccinated farms, check, sequence, whole genome, your strains, particularly if you are having an outbreak or you are having inclusive losses related to PRRS. That's all. Thank you very much, and sorry for the break.